Well, assalamu alaikum. Namaste,
Good
guys.
morning, sir.
Good morning. Good afternoon.
Sir, good morning. Good morning, sir. Good morning, sir.
Good morning.
Subratri, sir.
What about Microsoft Teams? some of you also has a problem with Microsoft Teams. No? Okay, let's start, guys. As you see, our logo of our department, we teach you for life, not for exam. And today, our topic is the muscle contraction. Really, it is last lecture, fifth lecture. But previous lectures, two lectures, were at our weekends and festivals. So today I deliver for you our fourth lecture, and next week will be last lecture in this semester about nerve fibers. Okay, today the muscle contraction. When most people think of muscles, they think of the muscles that are visible. Yes, do you agree? Just under the skin. Yeah, muscles under the skin, particularly of the limbs, and these are skeletal muscles, so named because most of them move the skeleton, our skeleton. But there are two other types of muscle in the body, which with distinctly different jobs. I think you know these types. Okay, what types? Two types. Skeletal muscles. What else? Two types of muscles. Smooth muscles. Good for you. And cardiac muscle. Thank you. Yes, of course. Cardiac muscle are found in the heart. This muscle concerned with pumping blood through the circulatory system. Smooth muscle is concerned with various involuntary movements, such as having one's hair stand on, and when cold or frightened, or moving food through the digestive system. Smooth muscle. Okay, this chapter will examine the structure and function mainly of skeletal muscles. Okay, mainly of skeletal muscles, about skeletal muscles. And after studying this chapter, you will be able to describe the organization of muscle, specify the functions of skeletal muscle tissue. Then we will identify the structural components of a sarcomere. Then explain the key steps involved in the contraction of a skeletal muscle fiber. Compare the different types of muscle twitch, and examine exercise and muscle performance. Okay, and our first question, okay, sir. Our first question is the organization of the tissue muscle as tissue level. Muscle is one of the four primary tissue types of the body. And the body contains three types, as we said, of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. All three muscle tissues have some properties in common. They all exhibit a quality called excitability. If you watched previous lecture about action potential, now you know what does it mean. Excitability, as their plasma membrane of muscles can change their electrical state from polarized to depolarized, and send an electrical wave called an action potential along the entire length of the membrane. While the nervous system can influence the excitability of cardiac and smooth muscle to some degree, Skeletal muscle completely depends on signaling from the nervous system to work properly. On the other hand, both cardiac muscle and the smooth muscle can respond to other stimuli, such as hormones and local stimuli, internal stimuli. So, as we said, the body contains of three types of muscle, and first type is cardiac muscle. Here you can see cardiac muscle fibers, each have on 1 to 2 nuclei, purple color, and are physically and electrically connected to each other so that the entire heart contracts as one unit called a syncytium. Okay, next type is skeletal muscle. Skeletal muscle fibers are multinucleated. This is one skeletal cell. One muscle fiber is one muscle cell, multinucleated cell. Multinucleated structure that compose the skeletal muscle, and because the actin and myosin are not arranged in such regular fashion in smooth muscle, the cytoplasm of a smooth muscle fiber has a uniform non-striated appearance. There is no striated in this muscle. So, three types: smooth, skeletal, and cardiac muscle. Common feature is excitability, but different structure. Okay. So, functions of skeletal muscle. The best known feature of skeletal muscle is its ability to contract and cause movement. Skeletal muscle act not only to produce movement but also to stop movement, such as resisting gravity to maintain posture. Small constant adjustments of the skeletal muscles are needed to hold the body upright or balanced in any position, any posture, any position. Muscles also prevent excess movement of the bones and joints, maintaining skeletal stability and preventing skeletal structure damage or deformation. Joints can become misaligned or dislocated entirely by pulling on the associated bones. Muscles work to keep joints stable. Skeletal muscles also protect internal organs, particularly abdominal and pelvic organs, by acting as an external barrier or shield to external trauma and by supporting the weight of the organs. Skeletal muscles are located throughout the body at the openings of internal tracts to control the movement on various substances. These muscles allow functions such as swallowing, urination, defecation to be under our voluntary control. Skeletal muscles contribute to the maintenance of homeostasis in the body by generating heat. Muscle contraction requires energy, and when ATP is broken down, heat is produced in our body. This heat is very noticeable during exercises when sustained muscle movement causes body temperature to rise, and in case of extreme cold, when shivering produces random skeletal muscle contractions to generate heat by shivering. Skeletal muscles, organization of skeletal muscle tissue. Skeletal muscles are organs that contain connective tissue, connective tissue, blood vessels, nerves, skeletal muscle tissue, single skeletal muscle cells, as we said, muscle fiber equals muscle cell. Muscle fiber is muscle cell, multinucleated muscle cell. Muscle cell also called muscle fiber, as you know. Okay, because skeletal muscle cells are long and cylindrical, they are commonly referred to as muscle fibers. Skeletal muscle fibers can be quite large for human cells, with diameter up to 100 micrometer and length up to 30 centimeters, or approximately 12 inches, yeah? 11.8 inches. The plasma membrane of muscle fibers is called the sarcolemma. The cytoplasm is referred to as sarcoplasm, and the specialized smooth endoplasmic reticulum, which stores, releases, and retrieves calcium ions. This part called sarcoplasmic reticulum. Sarcoplasmic reticulum. Here you can see whole muscle as organ, whole muscle as organ, and here muscle fiber. Every muscle fiber is a big multinuclear cell, have many nuclei. Okay, and here you can see a closer structure of the muscle fiber. Yellow color is sarcoplasmic reticulum, where stores, release, and retrieves calcium ions. Sarcoplasmic reticulum, yellow. To better understand structure of myofibrils, let's watch the video, guys. Just a moment. Each muscle fiber itself contains cylindrical organelles known as myofibrils. Each muscle fiber contains hundreds or thousands of myofibrils, which themselves are bundles of proteins called actin and myosin. These run the length of the muscle fiber and are important in muscle contraction. Surrounding the myofibril, there is a network of tubules and channels called the sarcoplasmic reticulum, where calcium is stored, which is also important for muscle contraction. Each myofibril can be broken down into functional repeating segments called sarcomeres, which are the basis for muscle contraction theory, known as sliding filament theory. Okay, did you see this video? Okay, the sarcomere, the striated appearance of skeletal muscle fibers, is due to the arrangement of the myofilaments of actin and myosin in sequential order from one end of the muscle fiber to the other. Each pocket of this microfilament and their regulatory proteins, troponin and tropomyosin, is called a sarcomere. Sarcomere 1, sarcomere. Here, green color is actin and purple is myosin. Thin filaments and thick filaments of our muscle. Thin actin with troponin and tropomyosin, and thick filaments is myosin with special parts. Okay, we discussed about it later in this lecture. Okay, this is sarcomere, one unit of our muscle fiber. The sarcomere is the functional unit of the muscle fiber. The sarcomere itself is bundled within the myofibril that runs the entire length of the muscle fiber and attaches to the sarcolemma at its end. As myofibrils contract, the entire muscle cell contracts. Each sarcomere is approximately 2 micrometer in length with a three-dimensional cylinder-like arrangement and is bordered by structures called Z-discs. Here you can see Z-discs, also called Z-lines, to which the actin myofilaments are anchored. Here you can see actin, green actin, thin filaments of the muscle anchored or fixed by Z-lines. Fixed by Z-lines. Because the actin and its troponin-tropomyosin complex form strands that are thinner than the myosin, it's called the thin filament of the sarcomere. Likewise, because the myosin strands and their multiple heads projecting from the center of the sarcomere towards, but not all the way to the Z-discs, yeah, as you see, have more mass and are thicker. They are called the thick filaments of the sarcomere. Okay, thin filaments and thick filaments of the sarcomere. Here you can see closer the structure of a thin filament, actin filament, actin protein. Actin protein consists of includes troponin and tropomyosin or tropomyosin complex. Troponin, tropomyosin complex. This is thin filament of muscle fiber. And thick filament, the structure of a thick filament, myosin molecule or myosin proteins consist of myosin head that can pivoting, myosin hinge or heads hinge, and myosin tail. Here you can see closer organization of skeletal fiber or sarcomere, Z-lines, a stretched out sarcomere, Z-line, and thin filaments. Here thin filaments are red or orange, red. Here you can see M-line that fixed thick filaments, myosin filaments, and here also Z-line. Okay. Neuromuscular junction or muscle contraction. Signaling for a skeletal muscle fiber to contract, its membrane must first be excited. In other words, it must be stimulated to fire an action potential coming from the nervous system. Again, if you watched previous lecture on YouTube, action potential, and watched animation, you know that skeletal muscle fiber to contract must be stimulated by action potential that coming from nervous system or through nerve fibers from nervous system. Signaling begins when a neuronal action potential travels along the axon of a motor neuron and then along the individual branches to terminate at the neuromuscular junction. We will examine this question about neuromuscular junction, about synapse at our next lecture. Propagation of an action potential along the sarcolemma is the excitation portion of excitation-contraction coupling. Recall that this excitation actually triggers the release of calcium ions from its storage in the cells of sarcoplasmic reticulum. For the action potential to reach the membrane of the sarcoplasmic reticulum, there are periodic invagination in the sarcolemma called T-tubulus. T-tubulus. Here you can see T-tubule and two terminal cistern of sarcoplasmic reticulum is triad. Okay, T-tubule and couple of terminal cistern of sarcoplasmic reticulum is triad. The T-tubulus carry the action potential into the interior of the cell, which triggers the opening of calcium channels in the membrane of the adjacent sarcoplasmic reticulum, causing calcium to diffuse out of the sarcoplasmic reticulum and into the sarcoplasm. It's the arrival of calcium in the sarcoplasm that initiates contraction of the muscle fiber by its contractile units or sarcomere. Or sarcomeres. As long as calcium ions remain in the sarcoplasm to bind to troponin, which keeps the actin binding sites unshielded, and as long as ATP is available to drive the cross-bridge cycling and the pulling on of actin strands by myosin, the muscle fiber will continue to shorten to an anatomical limit. Here in this slide, in this picture, you can see an action potential arise at neuromuscular junction. Here, often transmitter of axon terminal is acetylcholine. Acetylcholine is transmitter or mediator. Acetylcholine is released, binds to receptors, and opens sodium ion channels, leading to an action potential in sarcolemma. Action potential travels along the T-tubulus, to sarcoplasmic reticulum, and then calcium releases. A cross bridge forms between actin and the myosin heads, triggering contraction. As long as calcium ions remain in the sarcoplasm to bind to troponin, and as long as ATP is available, the muscle fiber will continue to shorten. Sarcomeres will shorten, and all muscle fiber and whole muscle will shorten. Muscle contraction usually stops when signaling from the motor neuron ends, which repolarizes the sarcolemma and T-tubulus and closes the voltage-gated calcium channels in the sarcoplasmic reticulum. Calcium ions are then pumped back into the sarcoplasmic reticulum, which causes the tropomyosin to reshield or recover the binding site on the actin strands. A muscle also can stop contracting when it runs out of ATP and becomes fatigued. Guys, it's a very difficult, very hard question. So now I explain, then I show you some video and you will understand. Okay, relaxation of a muscle fiber. Calcium pumped back into the sarcoplasmic reticulum, as I said, which causes the tropomyosin of actin filaments to reshield the binding site on the actin strands. A muscle may also stop contracting when it runs of an ATP and becomes fatigued, and as I said, the sliding filament model of contraction, or microscopic mechanism of muscle contraction. When signaled by a motor neuron, a skeletal muscle fiber contracts as the thin filaments are pulled and then slide past the thick filaments within the fiber sarcomeres. This process is known as the sliding filament model of muscle contraction. The sliding can only occur when myosin binding sites on the actin filaments are exposed by a series of steps that begins with calcium entry into the sarcoplasm. When myosin, due to these heads, here you can see, bind actin filaments. When a sarcomere contracts, the Z lines move closer together, and the I band, this part, sorry. I band here, there is no myosin in this part called I band, just actin filaments, and the A band becomes smaller. The A band stays the same wide, the A. This is the A band, myosin band. In contraction cycle, A band stays the same wide, but I band becomes smaller. At full contraction, the thin and thick filaments overlap. Tropomyosin is a protein that winds around the chains of the actin, of the actin filaments, and covers the myosin binding sites to prevent actin from binding to myosin. Here you can see tropomyosin covers the myosin binding sites to prevent actin binding with myosin. Troponin also has a binding site for calcium ions. To initiate muscle contraction, tropomyosin has to expose the myosin binding site on an actin filaments to allow cross bridge formation between the actin and myosin microfilaments. This is first step of muscle contraction. The first step in the process of contraction is for calcium to bind to troponin. So that tropomyosin can slide away from the binding site on the actin strands. Okay, have you got it? Then head of myosin binds to actin. Active site exposure, here you can see slide away from the binding sites on the actin strands, tropomyosin, and here you can see there is no tropomyosin, but there is active site for binding head of myosin with actin. Third, next step, this process allows the myosin heads to bind to these exposed binding sites and form cross bridges. Myosin head binds to actin, active site of actin, where were tropomyosin's molecules. During the power stroke, the phosphate generated in the previous contraction cycle is released. This results in the myosin head pivoting toward the center of the sarcomere, after which the attached ADP and phosphate group are released. Myosin head binds and pivoting to the center of sarcomere, like this movement. A new molecule of ATP attaches to the myosin head, causing the cross bridge to detach. The myosin head hydrolyzes ATP to ADP and phosphate, which returns the myosin to the cocked position. At last, cross-bridge formation occurs when the myosin head attaches to the actin, while adenosine diphosphate and inorganic phosphate are still bound to myosin. Okay, in order to better understand this mechanism, let's watch the video, guys.
Control. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, Tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay. Types of muscle contractions or types of muscle twitch. In isotonic contraction, there are two general types. In isotonic contraction, where the tension in the muscle stays constant, a load is moved as the length of the muscle changes, muscle shortens. There are two types of isotonic contractions, concentric and eccentric. A concentric contraction involves the muscle shortening to move a load. An eccentric contraction occurs as the muscle tension diminishes and the muscle lengthens. An isometric contraction occurs as the muscle produces tension without changing the angle of a skeletal joint. Isometric contraction involves sarcomere shortening and increasing muscle tension, but do not move a load, as the force produced cannot overcome the resistance provided by the load. Exercise and muscle performance. Muscle cells can change in size. New cells are not formed when muscles grow. Instead, structural proteins are added to muscle fibers in a process called hypertrophy. So cell diameter increases. Here. Increases. The reverse, when structural proteins are lost and muscle mass decreases, is called atrophy. A related muscle atrophy is called sarcopenia. Cellular components of muscles can also undergo changes in response to changes in muscle use. Sorry, guys. Endurance exercise. Slow fibers are predominantly used in endurance exercises that require little force but involve numerous repetitions. The aerobic metabolism used by slow twitch fibers allows them to maintain contractions over long periods. Long-distance runners have a large number of slow oxidative fibers and relatively few fast oxidative and fast glycolytic fibers. And resistance exercises as opposed to endurance exercise require large amount of glycolytic fibers to produce short, powerful movements that are not repeated over long periods. So, and in the end of our lecture, some short movie if you want. If you don't want, Bye-bye, and write down me your questions, please. Okay, guys?
Yes, sir.
Can you show me your faces? Achint, how are you doing? Fine. What about online study, online education?
It's going on, sir.
Yesterday said that you will study offline, but today, today they said that you will continue online education.
So our first semester will be online.
What the weather like? What the weather like in India, in Kashmir, Rajasthan? What the weather like?
Kashmir.
In
So
Kashmir,
Kashmir is old.
it's
So
hot.
cold that it was one of the coldest nights in the 27 years.
Mm.
8 years.
8 or 27, something like this.
In
8 years. It was
in
-8 last night.
in Astana, minus 30 degrees, minus 30, minus 31 degrees.
Sir, in Kashmir actually,
So,
-4 of Kashmir is much colder than -30 of
Exactly.
because we don't have anything
Yes,
like
sir.
heating systems here.
means
The wind disturbances are, actually
it's interesting.
And the development, like there are many things in Kazakhstan. In Kashmir, we don't have those equipments like that eternal reset
Exactly.
system. And
Yeah.
we are just with that traditional one thing known as Kangdi.
Very poor for rabbits.
Okay, our time is out, 12:50. Thank you, guys. If you have some questions, please write down to my chat, my private chat, or our general chat, okay? Thank you.
Okay, sir.
Bye-bye.
Okay, sir.
Assalamu alaikum. Bye-bye.
Bye, sir.